Hello everyone, welcome to our YouTube channel Applied Forensic Research Sciences. I am Shruti Karnar, volunteer at AFRS, here with a new YouTube video on the topic double helix structure of DNA. In this video, we are going to discuss about the introduction of double helix structure, then the history, definition, structure, Chargovs Rule, diagrammatic representation of double helix structure, chemical nature, then characteristics followed by functions, and what is the forensic significance of double helix structure of DNA. To begin, first of all, let's discuss about the introduction of double helix structure of DNA. So when we are saying about the double helix structure of DNA, what comes in mind first is about the DNA, that is deoxyribose nucleic acid, which is the hereditary material in humans and almost all other organisms, in which all the genetic information is carried from one generation to another generation of that specific species. And the human DNA is consisting of about 3 billion bases, and more than 99% of those bases are same in all people. As we have got known with the basic idea of double helix structure of DNA, so now let's discuss that what was its discovery or how it was founded and what was its structure in further video. So when discussing about the history of the double helix structure of DNA, the first name which comes in mind is Friedrich Miescher, because he firstly isolated the DNA from the pus cell. Later on, in 1933, while studying the virgin sea urchin eggs, it was found that DNA is found in the nucleus of the cell by John Bratchett. Followed by him, William Osbury in 1937 showed about the regular structure of DNA using the X-ray diffraction patterns. Then Avery, MacLeod and McCarthy discovered about the transforming principle of DNA and identified DNA as the transforming principle from one generation to another in 1943. Then it was the time for the discovery of double helix structure of DNA, which was given by Francis Crick and James Walton in 1951 within the University of Cambridge at the Cavendish Laboratory, and observed the genetic material DNA and introduced it in the form of double helix structure. Also, it is known as Watson and Crick model of DNA. As we have discussed in the introduction pattern, that is, DNA is also known as a genetic material, and according to the history, Watson and Crick give the double helix structure of DNA. So what it can be defined as? So DNA is a self-replicating material that is present in nearly all living organisms as main constituent of chromosome. It is carrier of genetic information. So what does DNA carry is genetic information which is present on genes present over the chromosomes. So DNA is self-replicating material, because from one DNA we can get the multiple copies of DNA which is present in all living organisms. As we have discussed already about the definition introduction, so now let's get idea that what is the structure of DNA. So DNA, it is made up of two polynucleotides chain which are coiled around each other to form the double helix structure, which carries the genetic instruction which are useful for growth, development, functioning and reproduction of all organisms. As we have discussed it earlier, that DNA is the genetic material present in all organism. So how it is made, that is two polynucleotide chain which are round around each other, and they act as a double helix structure which are added upon by the nitrogenous bases placed centrally, holding together two strands. These four bases are adenine, guanine, cytosine and thymine, that is ATGC, adenine, thymine, guanine and cytosine. These four bases are attached to each other by the sugar moiety. As we have discussed earlier, that DNA is made up of two polynucleotide chain, so it can also be called as polymer because it is made up of smaller units known as polymer, which in turn forms a chain of nucleotide bases and then forming the double helix structure, which is linear and long. So this both strand or both chains are coiled around each other, are united with the help of bonds between the bases of adenine, forming the base pairs with thymine and cytosine pairing with guanine. That is, A is pairing with T and G is pairing with C, forming AT and GC pairs. These chemical bonds are referred to as sugar phosphate backbones because they are formed by phosphate and sugar molecules. In summary, the double helix structure is made up of two strands which are coiled around each other and having the base pairing of adenine, thymine, guanine and cytosine, hence called as sugar phosphate backbone, forming of phosphate and sugar molecules. As we have earlier discussed about the base pairing of AT and GC, based on that, Erwin Chargoff in the time period of 1905 to 2002 gave the two basic rules which are dependent upon the DNA molecule, a double stranded structure. So this is first rule is A percentage equals to T percentage and percentage of G is equals to percentage of C. That is how many percent of adenine is present, same percentage of thymine will be present. This is validation of the rule constitutes the basis of Watson and Crick pairs in double helix model. The second rule is about the A percentage and T percentage and G percentage and C percentage, which holds for the each strands of two DNA strands, and this is used to describe only a global feature of base composition in a single DNA strand. As we discussed earlier about the base pairing of adenine, thymine, guanine, and cytosine, also amongst the Chargovs rule, so is the given in the above diagram. This diagram depicts about the AT and GC pairing, where the blue strand is showing about the DNA, which are nucleotides of monomer, which are made up of three basic components: a nitrogenous base pair, a pentose sugar called as ribose, and a phosphate group. This pentose is having the meaning derived from the word 5 carbon. The pink band shows about the DNA made up of adenine binding with thymine by two hydrogen bond. These two strands are combined together to form a double-stranded double helix structure of DNA. So as next is about the gray part. Gray part is showing about the G and C pairing, that is guanine and cytosine binding. Length of one full round is shown to be 3.4 nanometer. Till now we have completed about the definition, structure, composition. Next part to be completed is about the chemical nature. Chemical nature of DNA is that is DNA is negatively charged micromolecule. The negative charge present on the DNA is due to the presence of phosphate group, and the phosphate group in DNA, it is made up of negatively charged or ionized oxygen atom. So whole DNA becomes negatively charged due to this small oxygen atom present over the phosphate group, hence phosphate group gets negative and hence the DNA is also negatively charged micromolecule. As we say, DNA is a genetic material. So for being a genetic material, it has to need some important characteristics. Those characteristics are: it must contain complex biological information in stable form. Also, it must replicate faithfully. So is the DNA, that is, it is self-replicating. It must encode phenotype, and it must also be capable of variation, and these all qualities are present in DNA. So DNA is a complex molecule which can replicate, having capability of variation, and it is having stable biological information which codes for various genes. Functions of DNA: DNA is involved in various works such as translation, transcription, replication, gene expression, and mutation. That is, in the RNA and DNA, there occurs a translation and transcription process. Also, for change in genetic expression, there is mutation. It is helpful in expression of genetic variations in various individuals or transferring the genetic material from one individual to another individual, and it can replicate itself. Whatever earlier we have discussed are the importance or DNA functions in general life, but what is the forensic significance of DNA? So in forensic science field, DNA plays a vital role because it is used to prove innocent and convicting the guilty. It is also used in the identification of person because of acting as a biological evidence as it is transferred from one crime scene to another. For example, the hair sample, it can be used for the identification of DNA, and it can also be further used to identify the person. This biological evidence we get in form of various swabs such as swab from buccal cavity, saliva, semen, blood, vaginal swab as a touch DNA. So from this information we can code for various genetic material and then we can identify the person. With this short and sweet discussion, I would like to conclude my video. Thank you.